Good evening. I hope I'm audible. So, okay, let's just wait for 5 more minutes. If anybody else joins, then we can initiate the topic.
Ma'am, you are not audible. I think your mic is on.
Yes. Yes, I was just checking the presentation. Yeah, I think it's visible and I'm audible, right?
Yes.
Okay. Yeah. So, in today's session, till now we have discussed about the gene editing technologies, right? So, gene editing technologies. For example, we have discussed zinc finger nuclease, we have discussed the TALEN technology and also the CRISPR technology. All these technologies are being used for the gene therapy, for the modifications, for to treating the diseases. Okay. So from there only, we today will be discussing three different diseases, which are the genetic disorders, okay, and which are being treated nowadays using this gene editing technologies. Okay. So the three genetic disorders we'll be discussing today, that includes thalassemia, okay, severe combined immunodeficiency, and hemophilia. Okay. So hmm, okay. Okay. So first disease that we have is thalassemia. Okay, no problem. So thalassemia is a disease that is related with the blood disease. Okay, so in case of thalassemia, we know that blood has function to carry the oxygen throughout the body. Okay, and that it does with the help of hemoglobin. Okay, hemoglobin is a protein that is associated or present in these red blood cells. So this hemoglobin is made up of several subunits. Okay, that it attached with the heme group. That means iron containing heme group. So it is a hemoglobin protein. Okay. So these globin genes are expressed differentially as different stages of the development and produce hemoglobin. Okay. So there are two chromosomes, chromosome 16, chromosome 11, where these globin genes are present. Okay. So here you can see that there are several globin genes associated, alpha 2, alpha 1, then here also gamma, uh, delta, beta. So on chromosome 16 and 11, these genes are present. So during the embryonic phase, here you can see in the embryonic phase, so we have this these three different variants of the embryonic hemoglobin. Like during the when the embryo is there, these three forms or these three units of globulin protein form together and make a active hemoglobin protein. Okay, so that includes these zeta 2 and epsilon 2. So epsilon is coming from this chromosome 11, that being coded from chromosome 11, and zeta is being coded from chromosome 16. Okay, and both of two of these subunits are combined together and making the embryo hemoglobin. So these are three different hemoglobins that are formed during the embryo. When the embryo develops into fetus, then the globulin subunits are changed. Okay, so here the hemoglobin subunits in the fetus is alpha 2 and gamma 2. So alpha is being coded from these either of these two genes present on chromosome 60, alpha, along with the gamma gene that is being coded from these two exons present on chromosome 11. So these two make alpha 2 gamma 2, which is the fetus hemoglobin. And then when the child birth happens, and then the adult, in the adult form, these two forms of hemoglobins are present. Okay. So these are two adult hemoglobin where HbA, which comprise alpha 2 beta 2, this one. So beta is coming from chromosome 11, alpha is coming from chromosome 60. Alpha 2 and beta 2, they make the HbA, hemoglobin A, which is the 95% of the total adult hemoglobin. And hemoglobin A2, HbA2, which is alpha 2 and delta 2, that is the remaining 5% portion of the hemoglobin. Okay. So here, this particular uh, units like in this case, two subunits of alpha and two subunits of beta. Okay, they make the active hemoglobin HbA, which carries the function of oxygen transport in our body. Okay. Okay. So when there is any of these subunits are absent, okay, for example, you need total 2 alpha and 2 beta. Okay. If you have only 1 alpha and 2 beta, then the active hemoglobin will not be formed. Okay. In that case, there will be problem in blood carrier, RBCs synthesis, and everything. So that is how this is entire disease is the reduced production of the subunits of the hemoglobin. That includes alpha and beta, and that is how the types of thalassemia is given. Okay. So depending on the type of polypeptide chain whose synthesis has been impaired, the type of thalassemia is classified. Okay. So main categories of thalassemia is alpha thalassemia, beta thalassemia, and delta beta thalassemia. So alpha thalassemia, in that case, absent or decreased production of alpha globulin chain. In beta, we have either absent or reduced production of beta globulin chains. In delta and beta, reduced production of both delta and beta globulin chains. Only and then we have the rarely occurring thalassemia that includes only delta thalassemia and gamma thalassemia, decreased production of any of these uh, globulin chains. Okay. So as so the most common type of uh, that includes these alpha and beta among these three. Okay. So in case of alpha thalassemia also, so we have different types. For example, so um, as we are diploid organisms, we have two homologous chromosomes. Okay, so we have two alleles of the chromosome. Okay, so in our if at one chromosome the alpha gene is intact, the subunit that codes for the alpha gene is intact. However, in from other gene it is deleted. Now, so from this chromosome you will get the alpha production, but from this one you will not get the alpha production. So that is one type of alpha thalassemia, alpha and minus. Okay, and then we have another thalassemia which is alpha O. That means deletion of alpha gene from both of the alleles of the chromosomes. Okay. So in fetal life, the homozygous type of alpha 0, so this thalassemia is minus minus, leads to an excess this gamma chain and formation of gamma 4 tetramers. Okay, these are referred as hemoglobin Bart. So this condition known as hemoglobin Bart's hydrops fetalis is associated with intrauterine or neonatal death. Okay, so further abnormal hemoglobin composed of 4 delta globulin chain has been detected in 2 individuals with rare HbH disease. Okay, so these are the some rare diseases. So as we have seen in fetus, we have alpha 2 and gamma 2. So if we don't have this alpha, if we have alpha 0 mutation, then only gamma subunits will be produced that will make only gamma uh, tetramer, and that will be referred as HB Bart's hydrops fetalis. Okay. And in alpha thalassemia, there is so these are a genetic disorder, and this comes under the autosomal recessive disease. Okay. So that means they are not carried on the sex chromosomes. However, they are carried on the other remaining chromosomes. And that is an recessive disease. That means in um, both the copies of the faulty genes are need to be present in order for the condition to be expressed, in order for the disease to be expressed. So it is a recessive disease. Two copies are required. However, there is also one alpha thalassemia mental retardation, which is an X-linked disease. Okay, so a type of alpha thalassemia known as ATR16, which is alpha thalassemia intellectual disability syndrome, also characterized by mild to moderate mental retardation. So this ATR16 syndrome is called by deletion of a part of chromosome 16. A deletion of a part of chromosome 16 here from P13.3, a band on the short end of the chromosome, to the end of the chromosome. So this can either be due to balance translocation or a de novo deletion, and the genes affected include hemoglobin alpha 1 and the hemoglobin alpha 2. So this alpha thalassemia actually intellectual disability is a subtype of ATR16 syndrome result from ATRX gene mutation. Okay, so that means the mutation is there and ATR and X chromosome, but there are certain translocation uh, or de novo deletions has taken place because of this. That also includes the deletion on the chromosome 16 as well. So altogether it is referred as ATR16. So in ATR16, the part of chromosome 16 that includes the alpha polypeptide of the globin chain is also being deleted from that chromosome, and that is how it is altogether characterized as X-linked alpha thalassemia, which shows a moderate mental retardation symptom. Okay. Then we have beta thalassemia. Okay. So similarly, beta thalassemia also, it is also caused by several mutation in the beta gene. Like some mutation has happened that affects the uh, false or incorrect beta gene, or there is a mutation that causes the making a stop codon in the gene of interest, and that is how the entire beta gene is not able to produce. Okay, so more than 350 disease causing mutation have been identified so far worldwide. So majority of the beta thalassemia phenotypes are due to mutation in one or more nucleotide in the beta globulin cluster or instant flanks that result in shift of the reading frame. Okay. Gross gene deletion beta thalassemia were reported in rare condition. Okay. For example, like there we have read that alpha 0 mutation, like deletion of the entire alpha gene. But in this case, the gross gene deletion is rare. Okay. So beta thalassemia mutation may occur in exonic, intronic, or promoter sequences. All type of missense, minor deletion, frame shift mutation result in hyper unstable beta chains. So mutation producing the typically recessively inherited form of beta thalassemia located in exon 1 and 2. So this beta genes having exons, 3 exons. So that majorly includes exon 1 and 2. And in exon 3, mutation in the phase termination codon result in dominant beta thalassemia in contrary to the classically recessive inheritance of beta thalassemia. So in beta thalassemia, some mutation cause beta globulin gene to downregulate, which are known as cis and trans-acting mutation, leading to downregulation of beta gene expression. Okay, so for example, like if there is a mutation that is present in the gene itself, and because of that mutation, if there is a less production of that gene, that is a cis acting mutation. However, if the mutation is present in some other locus or some other protein that affect the production of our gene of interest, that is known as transacting mutation. For example, this GATA1, transcription factor 2H, KLF1 are known as transacting mutation, while mutation in alleles of the beta globulin gene locus are known as cis-acting mutation. So these are the regulatory factor, transcription factors for the beta genes. That is why the mutation in these affects the production of the beta gene as well. And that is why it is a transacting mutations. And if the mutations are there in the gene itself, for example, deletion of the beta gene, point mutation, large deletion, that is the cis-acting mutations. Okay. So, yeah. So now comes the gene therapy. Okay. So in gene therapy, as we know that genetic disorders are there. So this is the beta gene that is having three different exons, exon 1, 2, and 3. Okay. So you can employ the uh, gene editing technologies. Okay. Okay. So now you know that your gene of interest is having a mutation. Okay. It is having a mutation here. Okay. So what you can do now to treat the patient, you can do one in vivo approach. In vivo approach is if the patient is lacking the nutrient, like for example, in case of our thalassemia patient, they're having a kind of anemic situation. Okay. uh, False production of the hemoglobin subunits, okay, that is causing the decrease in the RBC, okay, and the oxygen transfer. So you can just directly insert the correct copy or the correct gene or the corrected substituent that is lacking. For example, blood transfusion can be done. So the patients for that are affected with thalassemia, blood transfusion is one of the techniques that is being used. Like uh, a compatible blood is being given that is fulfilling the work that is not being done by their own blood. Then we have the ex vivo technology. In ex vivo, what we can do, you can apply the genetic engineering here. For example, you can take the hematopoietic stem cells, and then you transfer the corrected copy of the gene in that corrected copy of the gene in the stem cells here. Allow the stem cells to grow. Select the positive cells that have the corrected copy of the gene, and that stem cells you put it in the patient. Okay. So now when these corrected hematopoietic stem cells will be uh, transferred to the patient, then the correct gene copy will start making, because now the cells that are being placed in the patient is the correct harboring the correct gene, and that is how the patients will be treated. So similar approach here I am showing is a CRISPR-Cas9 based approach for the correction in the beta thalassemia mutation. Okay. So at the so here you can see this is the faulty locus where this is a beta 039 mutation, C to T mutation. Okay. So here you can see from C it has changed to T. Okay. It has changed to T. The nucleotide has changed from C to T. Okay. So what we can do using the CRISPR technology, we can make a break at a desired locus, which is our desired locus is now here. Okay. So Using the CRISPR technology, what we have to do? We have to have a Cas9 protein that will make the cut, and to guide the Cas9 protein, we need to have a guide RNA. Okay, and now we know the gene sequence. From there, we can make or design the guide RNA. Okay, so this is the guide RNA that has been designed, which is in fusion with the tracer RNA, which is binding to the Cas9 protein. So you can see that this is the guide RNA that has been designed, which is come here and bound to this faulty locus. Now the cut has been made here. Okay, so it will make a cut here. Now this is the template of the correct donor template of the correct sequence has been provided, where in place of T, you can see the corrected base, that is C, is present. Now, after making the cut, this machinery needs to be repaired. Okay, so yeah, you can also see like the guide RNA that we design. So this is an entire sequence. You can see the exon 2 starting from here, this ATG gene. So ATG is the start codon. So it's starting from here, and then from this sequence, the guide RNA has been designed. And this is the PAM sequence, AGG, which is like PAM sequence is NGG, right? So the PAM sequence is here. So when and you give this guide RNA with the help of PAM sequence, here you can see NGG, the Cas9 will be guided here and with the guide RNA in a PAM sequence, and we'll make a cut in this particular strand where the PAM sequence is present. So both the cuts will be made. Okay. So this CRISPR-Cas9 system binds by the CRISPR RNA, this CRISPR RNA, on the exon 2 of the beta globulin gene, cutting the DNA near the PAM sequence, cutting the DNA near the PAM sequence, causing the formation of a double-strand break and inducing the cell to activate the repair mechanism, such as homologous recombination. Okay, so the cell will use the single-stranded DNA, this template, donor DNA, as a template to repair the damage, and this allows the insertion of the correct nucleotide sequence instead of the one containing the beta 39 stop mutation. Okay. So if in this strand where we have given the corrected sequence, this will be used as a template to repair this DNA. And when this will be used as a template, the T will be removed and the correct base, which was should be there at that place, C, will come. And now this particular uh, exon is corrected and produce a correct form of the beta G. Hmm. So then we have other diseases that include severe combined immunodeficiency disease. So, yeah. So these diseases, which are severely combined immunodeficiency diseases, it has several mutation. Okay, different kind of mutations uh, gathered, and that cause altogether a severely combined compromised immune system. Okay, which is a defective humoral and cellular immune system in the organism. It is both the type which is autosomal recessive and the X-linked. Okay, because there are several mutations are reported. Okay, because of this, because of those mutation, the patient show compromised immunity. So the compromised immunity, you have the lower production of either T cells, B cells, and natural killer cells. Okay, so there also we have several subtypes. So first is this T minus B minus. So in T minus B minus, that means neither T cells or B cells are being produced uh, in this kind of disease. Then we have T minus and B plus. That means T is completely absent and B plus means excess production of the B cells, okay, or the B immunocells. So in T minus, B minus also we have subcategories. Oh, okay. So these are the mutations that have been given, or the genes that are mutated. So in T minus, B minus, we have T minus, B minus, or NK minus, means all the immune cells like T cells, B cells, and natural killer cells all are absent here. That is the adenine deaminase enzyme deficiency. Okay, and then we have T minus, B minus, but the natural killer cells are present. Okay, so here the RAG1, RAG2 deficiency, DNA PKC's enzyme, these are all the deficient enzyme. Okay, and then we have in this one where the B cells are being produced, however T cells are not being produced, but one case where natural killer cells are present and absent, and another one natural killer cells are present. Okay, so here also we have several subcategories, like for here IL-2RG, this particular receptor is uh, absent. Because of that, the maturation of the T cell and B cells is not happening. Here ILR7 uh, receptor is absent or defective. So these are the several uh, array of the genes that is being affected, that leads to severely combined immunodeficiency disease. Okay, so defective survival of hematopoietic lineage precursor. So this one, adenyl adenylate kinase are responsible in cellular mitochondrial energy homeostasis. Okay, so this adenylate kinase catalyzes the reversible transfer of a phosphoryl group from ATP to AMP. So from ATP to AMP, so this ATP, ADP, AMP concentration maintained within tightly regulated range is crucial for cell survival. So adenylate kinase 2 gene located on this particular chromosome contains 9 exons, and biallelic mutation in the AKT gene clusters causes reticular dysgenesis. Okay, dysgenesis, which is an autosomal recessive disorder characterized by early myeloid lineage differentiation arrest and impaired lymphoid development. So around 2% of the children with SCID are caused by reticular dysgenesis, okay, which is a biallelic mutation in this adenylate kinase 2 gene. Okay, so single nucleotide substitution, frame shift mutation, and large intragenic deletions are reported in patients with RD. It manifests to cause lack of innate and adaptive immunity, and 21 diverse type of recessive mutation have been reported in case of RD. Absent of granulocyte, lymphocyte, uh, hypoplasia of the primary and secondary lymphoid organs, and fatal sepsis within days after the birth are observed in the patients. Okay, so only 2% of the children with the SCID are caused by the RD. Then we have also X-linked SCID. So one of the most frequent SCID arises due to cytokine signaling abnormality is X-linked SCID. So here this IL-2RG gene present on the long Q arm of the X chromosome. Okay, at position 13.9 encodes cytokine receptor common subunit gamma YC. This particular, okay, this particular subunit, also known as interleukin 2 receptor subunit gamma or IL-2RG, IL-2RG or gamma C. So the common subunit gamma chain, gamma C, a transmembrane protein, which is a transmembrane protein, which is common component of the receptor of IL-4, IL-7, all these interleukins. For all these interleukins, it is a common receptor and essential for the development of T lymphocytes and natural killer cells respectively. So the gamma C also activates intracellular tyrosine kinase JAK3 for signal transduction to carry out cell growth and control of hematopoietic cell development. So mutation in this gene causes X-linked SCID, which accounts for 50% of all the SCID cases. So it is accounts for the 50% of the severely combined immunodeficiency, X-linked SCID. Okay, so this is also one of the schematic representation of gene therapy protocol used in X1 linked clinical trials. So CD34 positive hematopoietic stem cells are sorted from bone marrow because there are the hematopoietic cells are present. Okay, okay, and cells are transduced with specific vector containing the corrective human interleukin 2 receptor G gene, IL-2RG gene, and a period of culture. Okay, so these cells are harvested, activated, and cells are transduced with the vector containing the correct copy of this IL-2RG gene. So the gene corrected CD3 34 positive cells are reintroduced in the patient with or without conditioning that will result in curing the patient. So these are the genetic therapies that has been included, and that includes the CRISPR, TALEN, and zinc finger nucleases that cuts or make a cut at the faulty gene. Okay, introduced a new copy or the corrected copy of the gene and then put it back in the patient and allow the new growth or the corrected proteins growth. Okay, similarly we have another disease which is hemophilia. Okay, so this hemophilia is also, it is a X-linked uh, disorder. Mhm. Hmm. So it is an X-linked disorder. So it involves three phases. So as the as if it is a cut is made, that is being clotted. So if there is a defect in the clotting factors, then there will be no clotting of the blood. So the blood clotting also involves several steps: initiation, amplification, and propagation. Okay, so here are certain factors. So intrinsic factor, along with collagen we have factor 12. Okay, that will activate the factor 11, that will ultimately activate the factor 9 here. Okay, so the factor 9 activates the factor 10. Okay, and factor along with the factor 8, and that will make the active thrombin from prothrombin, and fibrinogen which will make the soluble fibrins, and stable fibrin that will make the blood clot. Okay, so this intrinsic pathway is initiated by this 12, which will be activated as 12A. Okay, so here the key proteins present here are the protein 9 and protein 8. So the coagulant factor 8 and factor 9 are very crucial factors in the blood clotting. Okay, and deletion and mutation in these genes. Okay, so here 9 and 8 all together activate the factor 10. Okay, so this all together, so this active factor 8, active factor 9, and the factor 10 all together, these three complex is known as factor XAs or 10As. Okay, which will make prothrombin into active thrombin that will leads to blood clotting. Okay, so if there is a mutation in any of these factors, the gene encoding any of these factors, then uh, there will be a disease which is called as hemophilia. Okay, so the gene therapy here being used, so here the gene the codon optimizer, the corrected gene has been synthesized and put it under the specific promoter, and with the help of adenovirus it is being delivered to the cells, and that is how the correct copy of the gene is being produced inside the patient. Okay, so similarly gene editing by adenovirus delivered zinc finger nuclease and donor DNA template. Okay, so this is the zinc finger nuclease being delivered here, making a cut there the factor, the correct factor, and the donor DNA is given. So zinc finger nuclease, we know 2 copies of the zinc finger are needed to make a double stranded cut. So here the double stranded cut is being made. After the cut is being made, because of the homologous recombination machinery, the correct factor will be added here, coded here. Okay, and this is the transcription and splicing. The mRNA fusion transcript is ready. So this mRNA translation and clotting factor secretion will happen. Okay, so now we'll see our questions. So a patient diagnosed with absent or reduced production of delta and beta globulin chains, which type of thalassemia does he has? Which one will be the correct option? Yes, if the both of the proteins or the globulin chains are absent, then it will be delta and beta thalassemia. Yeah, how many mutations have been identified so far worldwide known to cause beta thalassemia? We just now read that as well.
See,
Yeah, it is 350 mutations of beta thalassemia are known so far. Okay, yeah, which type of alpha thalassemia characterized as mild to moderate mental, characterized by mild to moderate mental retardation, and it is also an X-linked alpha thalassemia. Which one? It is ATR. Which one? ATR 16, the alpha thalassemia intellectual disability syndrome. And what was HB Bard syndrome? Alpha nil mutation in fetus. In fetus, where only like 4 delta copies are making the hemoglobins. That condition is a hemoglobin Bard syndrome. Okay, and here the ATR 16 will be the correct option for the mental retardation. Which of the following risks is not associated with blood transfusion therapy? Okay, so when the blood transfusion is happening, so allergic reaction and blood-borne infections are very common, and also similarly alloimmunization. However, the compromised immunity, that is not happening because uh, there is no such chance of the compromised immunity associated with the blood transfusion, because again it is coming due to the presence of the some of the mutation that is present on some genes, but that doesn't get carried with the blood. So the compromised immunity is not the risk that is associated with the blood transfusion therapy. So which of the following is not true about severely combined immunodeficiency? First one, a group of rare disorder caused by mutation in different genes. Is it correct? I told you SCID is a type of disorder that is due to mutation in different type of genes, like ADA genes, RAG genes, okay, adenylate kinase, AK2 gene. So these are the different genes, but all these are associated with this combined severe combined immunodeficiency, which is a group of rare disorders. Okay, so these are different disease caused by different genes, mutation in different genes, but they all are grouped together and referred as severe combined immunodeficiency. So the first option, whether it is true or not true, it is true. Mutated genes involve development involve development and function of the immune cells. Is it correct? The mutated genes are involved or related to the development of the immune cells. Um, don't take it like the um, let me just check it. No. So here it is saying that the genes that are being mutated here that are involved or related to the development of the immune cells. So that is correct, right? So the genes, like for example, these genes, they are just mentioning these genes are involved in the development of immune cells. So that is correct option, right? Only it is uh, what to say, in title is mutated that these genes, only this section of the genes it is being referred. Okay, so don't get confused with. So these mutated genes are involved in the development of the function of the immune cells. It is not saying that the after the mutation it is helping the development. No, that is not the meaning. I hope it is clear to you. Yeah, yeah, like these set of genes. It is kind of that, these set of genes. Then third is the X-linked SCID primarily affects female infants. So as it is an X-linked disease, so it will be its effect will be in the male progeny, right? Because an X and Y from the mother carrier it has taken. Okay, if it is like this, then however, in case of daughter, it will be like this. So daughters are always carrier in X-linked disease, not always, like mostly carriers, but in male, if it is an X-linked disease, in males, whenever they take this chromosome, they primarily affect the male infants. Okay, the word primarily is given. So X-linked SCID primarily affects the male infants, right? Not the female one. Because if this one we'll see, okay, so these four phenotype or genotypes you will be getting, right? But these two are healthy. However, among these two, this one become carrier and this one will be affected, right? So in this scenario, you can say that X-linked SCID primarily affects the male infants, not the female one. Okay. Then we have fourth one is SCID is inherited in an autosomal recessive pattern. Correct or not? Correct or not? Yes, this SCID is both autosomal recessive and also X-linked. So this is correct and this is incorrect because it primarily affects the male children. Okay. So what percent of SCID in children are caused by reticular dysgenesis? So reticular dysgenesis we saw, right? Here, adenylate kinase enzyme. Okay. Mutation in this gene causes reticular dysgenesis, which is characterized by early myeloid lineage differentiation arrest and impaired lymphoid development. And 2% of the children with SCID are caused by reticular dysgenesis. So the correct answer will be 2%. So low levels of which clotting factor caused hemophilia? Okay. So we have hemophilia A, hemophilia B, hemophilia C. Okay. So hemophilia A is associated with factor 8 deficiency, which is the classical hemophilia. Okay. And hemophilia B is associated with factor 9 deficiency, and hemophilia C is associated with factor 11 deficiency. So this one is the rarest form. Rarest form. These two are the X-linked disease. However, this hemophilia C is the autosomal recessive, and it is one of the rarest uh, factor or the type of hemophilia. So the layers of which of the clotting factors cause hemophilia. So option here is either factor 8 or 9, factor 12 and 11, only factor 10, and none of the above. So which one will be the correct option? A, exactly. So these are the most common classical type of hemophilia, which is caused by either factor 8 or factor 9. If you have factor 8 deficiency, you will be having hemophilia A. If factor 9 deficiency, hemophilia B. Correct option will be A. Okay, which of the following pair on type of hemophilia and its deficiency factor is correct? Okay, factor A. Uh, so hemophilia A, factor 9 deficiency. Correct or not? B, factor 9 deficiency. Correct or not? Hmm? Sorry, I didn't hear you. Yeah, this one is wrong. It is asking which one is correct. So A is wrong. What about B? Yeah, B is correct. C, which factor? So obviously all both are wrong. But so can you tell me hemophilia A, which factor deficiency it will be if not 9? 8. Yeah. B, we know 9. Factor C will be which one? 11. It's 11. Hmm. Okay, I have to check this. This one will come later. The intraseminal inversion. Okay, so which of the following characterize the T minus B plus severely combined immunodeficiency? Okay, if it is T minus B plus SCID, then what is the defect? Defect in both T and B? Sorry, no? Yeah. Then B, the absence of mature T and NK lymphocytes while B cells are present in increased number. So in T and yeah, so in T and B we have two types, right? T minus B plus. In one case we have NK negative, we have NK positive. So this particular one is talking about this one. Okay, this one, the absence of mature T and NK lymphocytes while B cells are present in increased number. This is correct. This is the absence of B cells. No, it is not. It will not be absence of B cell because it is B positive. And absent or decreased production of B cell, again, it is B positive, then this is not the correct one. This is all the question. I'll just tell you answer for this. Hmm. Severe hemophilia A. Okay. So, in case of hemophilia, for example, factor 8 in hemophilia A condition, factor 8 is the faulty gene, right? So there also we have several exons on factor in the gene coding for the factor 8. So in that only, if there is some intrachromosomal inversion involving the intron 22, so it has some 24 introns. Okay, so there is one scenario where you can read from the notes where the inversion is happening because of the similar sequence similarity. So due to inversion, intrachromosomal inversion here, so then again there is a changes and manipulation in this condition that changing the intrachromosomal arrangement of this around the intron 22. Okay, so that will result in the severe hemophilia A. Okay, so this one will be the correct option that involves the intrachromosomal inversion involving the intron 22. Okay, so that's it for today. Okay, so these are the questions. I hope it was clear to you. Okay, we'll see you again in next week. Okay.
Yes, sir.
Okay. Yes. Hmm. Tell me. Uh-huh. Sorry, I didn't understand your question. Can you repeat again? Guide RNA. Hmm. Hmm. Yeah, so here it is given that once the break is made. Let's see here. So once the break is made here, so here it is written, you can see here that the cell will use the single stranded uh, donor DNA. So in after making the cut, so you are in the cell, okay, in the cell you have the faulty gene here. You are inside the cells, what you are adding a sequence that will code for the Cas9 protein, uh, guide RNA that will make the guide RNA that will lead the Cas9 to the faulty gene. Okay, and then when with the help of these two, when the cut is made, you are also providing inside the cell one single stranded DNA. Okay, that single stranded DNA that you are providing. So once this help of Cas9 and guide RNA machinery, once the cut is made, once the here the cut is made, now to repair, so the cell will feel that there is a cut in our in the genomic locus, then the repair machinery will be active, and to repair it, this particular single stranded DNA or the donor DNA will come in the picture. So the cell's repair machinery will use this particular donor DNA which is having the correct nucleotide in it. Okay, so uh, so the priming will happen with the broken ends, single stranded DNA, and then with the cell's inbuilt repair machinery that includes the polymerase and ligases, the correct nucleotide, or the using this donor DNA, the DNA will be synthesized here. Okay, on that is how, or the correct nucleotide will be added. Okay, because you have made a cut. Okay, you have made a cut at the locus that is near the faulty gene. Okay, and then what you are providing a template that is having the correct nucleotide. So the repair machinery will be active. Maybe after several round of replication, you will get rid of the fault or the incorrect sequence completely, and the correct nucleotide will remain associated with it each round of replication, when each round of replication will happen. Okay, so suppose 50% population also get the correct nucleotide inserted in it, that will also start making functional protein, right? So that functional protein is now can be used to make the functional or the active beta gene and hemoglobin gene or chain. Okay. Okay. Yeah, either this, I have not employed this technology yet, or what is the easiest method will be, after the cut is made, you, instead of single stranded DNA, you can provide a correct DNA which is double stranded. Okay, which will be something like this. Okay. Okay, so a similar DNA which will be having homology with, suppose, this part, or like this part is homologous, the entire part, and then it will be inserted. So the correct copy will be inserted here. So here in the beginning where the C or other mutation was there, after the insertion of correct copy, the correct nucleotide will come here. Okay, so that can also be given. Yeah, yeah, you can say. Hmm. Okay, anything else? Okay, okay, see you next week then. Okay.